Okay, so shown here is an example of beta pleated sheet. Remember, sabi natin in beta pleated sheets, okay, we have here an almost completely extended peptide backbone. Now, what's the implication of that? The implication is we can form hydrogen bonds between different parts of a single chain, no, that doubled back on itself. Okay, meaning, okay, the backbone of the polypeptide chain is extended into a zigzag pattern which can be arranged side by side to form a structure, okay, resembling that of a series of pleats. Hence the name beta pleated sheets. Okay, so by the way, you have here your intrachain H bonding. Okay, so you can see from this figure, okay, you can see from the figure, it's a single polypeptide chain that doubled back on itself. Kumbaga, para siyang nag-U-turn sa pinanggalingan niya. Again, that's your intrachain bonds. Okay, you can see here the arrangement of your R groups, okay, alternate no, first above, then below the plane with respect, okay, to the plane. No, so from the same figure, you can see the zigzag pattern of your polypeptide chain. There. Now your beta sheets can actually what do you call this? Assemble in different ways. No, so you can have here an um anti-parallel, okay, arrangement wherein the alternating chains now run in opposite directions. That is, if the N terminus of the first portion of side chain is aligned with the C terminus of the second portion of the side chain. So there. You can also have here a parallel arrangement. If the peptide chain, okay, or if the peptide chains run in the same direction. Or in other words, if the chains are aligned in terms of their N terminal and C terminal ends. So there. Now focus your attention here, no? So here, the hydrogen bonds are between, okay, adjacent sheets containing your carboxyl and amino groups. So ayun, so 'di ba? So by the way, you can also have here pala H bonding between different polypeptide chains, no? So you call this your interchain bonds. Okay, let's move to the irregularities in the regular structures. Okay, there are other helical structures found in proteins. No? These are often found in shorter stretches than with alpha helix na alam natin. And they sometimes break up the regular nature of the alpha helix. Okay, so the most common here is the 3 sub 10 helix, now which has 3 residue per turn, okay, and 10 atoms in the ring, now formed by hydrogen bonds. Okay, same with 2 sub 7 and 4.4 sub 16, no, they follow the same nomenclature. Okay, so the second irregularity is your beta bulge. Now, so this is a common non-repetitive irregularity found in anti-parallel beta sheets. No, it occurs between two normal beta structure hydrogen bonds and involves two residues on one strand and one residue on the other. So here, okay, it means that two amino acid residues in one strand are hydrogen bonded in one amino acid residue from another strand. No? So, a beta bulge distorts the usual alternation of your side chains, no? Which causes a slight bend, as you can see in the figure, no, in the beta sheets, because again, two amino acids are bonded in one amino acid. Okay. Another irregularity is your reverse turn. Note that the protein folding, now, which we'll be discussing later in protein folding dynamics, it requires that the peptide backbone and the secondary structure should be able to change direction. Okay, so reverse turns marks the transition between one secondary structure and another. No, so for steric reason, okay, meaning to prevent R group crowding during turns, no, glycine is usually involved here. Remember, that the R group of glycine is only H. No, so you have a small R group, thereby less steric crowding, no, that can accommodate the looping of your chains. Another thing to note is that proline is sometimes involved in reverse turns, no, or your looping. Why? Because its cyclic structure can actually provide for the geometry of the turn. Okay, meaning it can introduce a sharp bend, okay, in the polypeptide chain, as can be seen in the figure. Okay, again, your loops actually connects helices, okay, and sheets, and they're often structured, no, but can be very flexible. Also, if you will notice, okay, so if you will notice the given description in the figure, there are different types of reverse turn, no? So you can just read on that. Okay, if you'll notice in the given figure, okay, the green line here, okay, so the whole of it is a single protein. Okay, and you can see that in this particular protein, there are different secondary structures. Okay, so you have your beta plated sheet and alpha helix and then some random coiling. Now, so we can say that the secondary structure is therefore observed in a localized portion of a protein. Now, what do you call this? No, the combination of different secondary structure. Okay, so you call that your super secondary structure, no, which we will be discussing on the following slides. Note that there are many different possible combinations of your secondary structures, no, as the polypeptide chain folds back, okay, on itself in a protein. So there. Okay? Again, as mentioned on the previous slide, supersecondary structure are the combination, okay, of the different secondary structures, no, provided and the following slides are the common feature of these. Okay, while common features of these, no, so while your domains here, on the other hand, okay, refer to the specific cluster of secondary structural motifs in proteins, and that protein domains are the conserved part of a given protein's primary and tertiary structure, no? So that can evolve and function independently. Now, so for example, okay, when proteins are denatured, okay, the part that can still exist and function is your domain, no, and it can evolve back to, or it can revert back to its original tertiary structure, a process we refer to as, okay, what was it, renaturation. Now, so we'll discuss more about this as we go along. Now, so your motif naman, okay, is the repetitive supersecondary structure. No, so this is the combination of your supersecondary structures. Now, example of which is your beta barrel, no, which is arranged from beta minders and Greek keys. Okay, so just a recap, no, supersecondary structures are the combination of different secondary structures, while motifs naman are the combination of supersecondary structure. There. Okay. So here are some examples of your supersecondary structures. A here is your beta alpha beta unit. Okay, so you have here two parallel strands of beta sheets connected by a stretch of alpha helix. B here naman is your alpha alpha unit. You have here, okay, two anti-parallel alpha helix. C naman, okay, is your beta minder. Here, you have an anti-parallel sheet formed by a series of tight reverse turns, no, connecting stretches of a polypeptide chain. Okay, so D naman here is your Greek key. This is a repetitive supersecondary structure formed, okay, when an anti-parallel sheet doubles back on itself. And then lastly, okay, you have here your beta barrel. No? Okay, so this is created when beta sheets are extensive enough to fold back on themselves. No? As mentioned on the previous slide. No? So this is an example of your motif. Okay, note that the supersecondary structures can be seen in almost all proteins with parallel strands. Okay, also note that the linkers or loops connecting different supersecondary connecting secondary structures, no, or supersecondary structures can vary in length. Okay, now let's move to the structure of collagen. So collagen is a protein that provides structure to much of your body and is a component of bone and connective tissues. No, its structure is organized, okay, in water insoluble fibers, okay, of great strength. Okay, so a collagen fiber, okay, consists of three polypeptide chains wrapped around each other, okay, in a rope-like twist or a triple helix, no, which is called as your tropo collagen. Okay, so focus your attention on this figure. Note that, okay, there are three repeating amino acid residues in collagen. Okay, we have here X here can be any amino acid and HYP. Okay, so HYP here is your hydroxyproline. Also note that the triple helix is arranged, no, in such a way that every third residue is inside the helix. And this is always occupied by glycine because it's the only amino acid, okay, small enough to fit. By the way, these three strands are held together by hydrogen bonds, okay, involving hydroxyproline and hydroxylysine. Okay, them. Huh. Also, okay, collagen is both intra and intermolecularly linked, okay, by covalent bonds, no, formed by the reactions of lysine and histidine residues, no. So the amount of cross-linking in a tissue increases with age. No, so this is the reason why meat from younger animals are tender, while those from older animals are tougher. Okay, so some trivia here, no? What if proline in collagen is not hydroxylated? Okay. Mangyayari niyan is you will have here a collagen that is less stable than the normal since you have less or no H bonding at all, no, between hydroxyproline and hydroxylysine residue. You'll develop here scurvy disease, no, with symptoms, no, such as bleeding gums and skin discoloration. No? So these are the results of fragile collagen. Now, if you will search the net, what is scurvy disease, it will tell you that this is a disease associated with lack of vitamin C. Now, why? It's because, okay, the enzyme that hydroxylates proline so as to maintain the normal state of collagen rather, requires vitamin C, no, to remain active. So note that um, we're among the species that cannot produce their own vitamin C, no? So that is why we need to supply it via our diet. I'm sure, no, with regards to scurvy disease, you've all heard the story, okay, about Ferdinand Magellan's crew, no? They have suffered here the said disease because since they're on their voyage, no, travel sila, they have limited food resources. And as a consequence, they're not getting the amount of vitamin C needed by the body, okay, to function well. No, so during this time, no, so they're not yet aware of the importance of vitamin C in their bodies. So hindi masyadong nag-show ng symptoms or naapektuhan si Magellan here, no, because by chance kay nakakain siya ng something na rich in vitamin C. So ayun. Okay, and now we move to the types of protein conformation. First, we have here fibrous proteins. So these proteins have an elongated shape which tend to have irregular and linear structures, and they also tend to aggregate together to form macromolecular structures. Okay, and they are mechanically strong and are insoluble in water and dilute salt solutions. Okay, they of course play an important structural roles in nature. There, and note that they also have a repetitive sequence. Now you can just read the given examples. Second, we have here globular proteins. Okay, so the peptide chain of these proteins are folded into spherical or globular shapes. Okay, so and they tend to be soluble in water and salt solutions. Now because of their polar side chains are on the outside and can interact with the aqueous environment via H bonding and ion dipole interaction, and most of their non-polar side chains are buried inside. Okay, note that they have different sequence or there's no pattern as compared with fibrous proteins. Now, so by the way, globular proteins here have chemical functions. No, para silang workers with specific tasks to do. No, like your enzymes. Okay, so this figure shows you the comparison of shapes of fibrous and globular proteins. So you can just check this. Okay, now let's move to the tertiary structure of proteins. So this is the three-dimensional conformation of a polypeptide or a protein. No? So this structure includes the conformations of side chains, which means that the side chains help in the stabilization of the structure, okay, position of any prosthetic groups, and arrangement of helical and pleated sheet sections with respect to one another. By the way, your prosthetic groups refer to the portion of proteins that do not consist of amino acids. Okay, note here that this structural level is dictated by the interactions between side chains. Okay, or to put simply, it's the interaction of R groups. So we can also say that the tertiary structure refers to the folded structure of a protein. Okay, so the importance of this level is that this is the structure, okay, at which the polypeptide chains become functional. So again, at this level, every protein has a specific three-dimensional shape wherein the side chains or the R groups are presented on the outer surface. Okay, what's the reason for this? Now, this allows for the interaction of the proteins with other molecules, no, which gives it its unique function. Okay, so here we'll talk about enzymes na and stuff. To reiterate, okay, the functions of a protein depends on its tertiary structure. And so it follows that when this is disrupted or denatured, okay, the protein loses its activity. Okay, so much about that, no? Let's move na to the forces involved in tertiary structures. Okay, so again, more of this later. Okay, no? So there are many types of forces and interactions that play a role in maintaining a protein in its correct native conformation. Okay, some of these forces are covalent, but majority of these are not. Okay, so we'll be discussing here no in on this slide about the forces and interaction involved in tertiary structures. First, we have here the covalent disulfide bonds. This is made from two cysteine residues from the same or different polypeptide chain. Remember, because sabi natin, proteins have more than one polypeptide chains. Okay? And also, if you still remember, we also mentioned from the previous lecture that cysteine, okay, likes to form bridges. Okay, so again, this is the covalent interaction. Note here that there are many methods available for the determination of the number and positions of disulfide links. Now, once you know the location of disulfide bridges, you can use that in combination with your knowledge in the primary structure of proteins to give the complete covalent structure of your proteins. Okay, so now we move to non-covalent interactions. We have here your salt bridges or the electrostatic interactions from your charge R groups. Okay, next naman, we have H bonding. Note that the H bonding here is different from that of the secondary structure. Because if you remember, it's the backbone hydrogen bonding that is the major determinant of the secondary structure. So when we're talking about the H bonding in tertiary structure, we're referring to the hydrogen bonds, okay, between the side chains, no? Specifically, your polar side chains. So by the way, note that the green line here is your polypeptide backbone. We also have here your hydrophobic and van der Waals interaction. Okay, between non-polar side chains. No? So in this figure, it's also shown here your metal ion coordination. There. This is particularly useful when we're discussing already about enzymes. Okay, so this is particularly useful when we're discussing na about enzymes and their cofactors. Pero ngayon pa lang, note that your cofactors are an essential non-protein component in enzymes so that activates them or plays a role in the chemical reactions, and usually your cofactors are metal ions. By the way, if this is not an enzyme, okay, then we can refer to the metal ion as your prosthetic group. Note here, in general term, no? So, note here that not all proteins necessarily exhibit all feature that we just mentioned or that we have just mentioned. Okay, so for instance, no, in myoglobin and hemoglobin, there are no disulfide bridges. Okay, so we'll discuss more about them, myoglobin and hemoglobin later.